Welcome to our channel Mootechie, exploring molecular biology. Today we will see about Carl Woese's three-domain classification system. This opening sets the stage for a deep dive into how we classify all life on Earth, moving beyond what we can see to what our genes can tell us. Why a three-domain system? The old five-kingdom system was limited because it grouped all prokaryotes into a single kingdom called Monera. Advances in molecular biology, however, revealed deep genetic differences that were completely hidden by physical appearance alone. The discovery of extremophiles, organisms thriving in extreme environments, challenged these old groupings and demanded an entirely new category. Ultimately, this new system aims to reflect the true evolutionary relationships between organisms based on evidence from genetic sequencing. This slide explains the why behind the shift. It wasn't just about adding another group, it was a fundamental change in how we understand life's family tree, driven by new genetic tools. Now, let's look at the big picture this system created, the phylogenetic tree of life. It's divided into three main branches: bacteria, archaea, and eukarya. Under bacteria, we see groups like cyanobacteria, filamentous bacteria, and gram-positives. Archaea includes organisms like methanogens and halophiles. Eukarya is the diverse branch containing slime molds, animals, fungi, plants, and various protists like ciliates and flagellates. This tree visually represents Woese's revolutionary idea. It shows archaea as a separate major branch, distinct from bacteria and more closely aligned with the lineage that led to us, the eukarya. The basis for this new tree was rRNA sequencing. Ribosomal RNA or rRNA is universal in all cells and evolves very slowly, making it an ideal molecular clock for tracing deep evolutionary time. Scientists compare the sequences of 16S RNA in prokaryotes or 18S RNA in eukaryotes across different organisms. The degree of divergence in these gene sequences is used to estimate phylogenetic distance and map out evolutionary history. This genetic method allows for a truly objective classification, moving us past reliance on just physical appearance or shape. This is the cornerstone of the entire system. By using a common, slow-changing molecule found in every living cell, Woese provided a consistent ruler to measure life's history. Here are the three domains at a glance. Life is now divided into three primary domains: bacteria, archaea, and eukarya. The rRNA phylogeny clearly shows that archaea are more closely related to eukarya than they are to bacteria. These groups are defined by distinct molecular synapomorphies, shared genetic traits, not just by their physical form. This system completely replaces the older, simpler view of just a prokaryote-eukaryote split. This summary emphasizes the core revelation. The deepest split isn't between cells with or without a nucleus. It's between two fundamentally different prokaryotes, with one being a closer relative. We have a simple visual here to reinforce the three domains. Archaea, labeled ancient or extreme, eukarya, labeled complete life, and bacteria, labeled true bacteria. A diagram shows three separate circles for bacteria, archaea, and eukarya, clearly distinguishing them. The image helps cement the three equal major categories in our minds, moving away from the idea of just bacteria and everything else. Let's explore each domain, starting with domain 1, bacteria. These are unicellular prokaryotes, meaning they lack a true nucleus or any membrane-bound organelles. A key distinguishing feature is that their cell walls are typically composed of a molecule called peptidoglycan. Their membrane lipids are built with ester-linked fatty acids, which form straight chains. The image shows a basic bacterial cell structure, highlighting the cell wall and the simple circular chromosome floating in the cytoplasm. This domain represents a vast diversity of life with critical roles in decomposition, causing disease, and applications in biotechnology like the oxygen-producing cyanobacteria. Here we get the defining biological details of true bacteria. The presence of peptidoglycan in their cell walls is a major chemical marker that sets them apart from the next domain we'll see. Now, domain 2, archaea. These are also prokaryotic in cell structure but are genetically and biochemically distinct. They lack peptidoglycan and possess unique cellular machinery. Their membranes contain special ether-linked lipids with branched chains made of isoprenoids which are much more stable. Many archaea are extremophiles, thriving in high heat as thermophiles, high salt as halophiles, or strong acid as acidophiles. Most importantly, genetic analysis reveals their ribosomal RNA has a closer affinity to eukarya than bacteria. Archaea break all the old rules. They look like bacteria under a microscope, but their chemistry is alien and often built for extreme worlds. Their genetic link to us is the key insight that reshaped the tree of life. Finally, Domain 3 Eukarya. These organisms are characterized by a true nucleus that houses their DNA and various membrane-bound organelles like mitochondria and chloroplasts. The domain includes the diverse kingdoms you are familiar with: protists, fungi, plants, and animals. Eukaryotic cells are structurally advanced with a complex internal cytoskeleton for support and movement, and their DNA is packaged into linear chromosomes. Their evolution is thought to involve endosymbiosis, where ancient bacteria were engulfed and became permanent, energy-producing organelles. This domain is our own. The complexity of the eukaryotic cell, likely born from ancient mergers, enabled the incredible diversity of visible life on our planet. The slide includes an anatomy of a cell image, likely showing a detailed eukaryotic cell with labeled organelles like the nucleus, mitochondria, and endoplasmic reticulum, contrasting it with simpler prokaryotic designs. This visual ties it all together, showing the structural complexity that defines eukarya and makes multicellular life possible. This slide offers a direct comparison between the old and new systems. On the left, the Five Kingdom system by Whittaker in 1969, based on morphological classification. It includes Kingdom Monera for all prokaryotes, Kingdom Protista, Kingdom Fungi, Kingdom Plantae, and Kingdom Animalia. This system lumped all prokaryotes together based primarily on physical traits and how they got their nutrition. On the right, the three-domain system by Woese in 1990, based on molecular phylogeny. It consists of domain bacteria, domain archaea, and domain eukarya, which itself contains the kingdoms protista, fungi, plantae, and animalia. The critical note is that this system split the old Monera because rRNA gene sequencing proved archaea are evolutionarily distinct from bacteria. Seeing them side by side makes the paradigm shift clear. It moved biology from classifying by what we see to classifying by what the genes reveal about shared history. Let's discuss the significance of the system. First, it redefined the tree of life. It fundamentally reshaped biological classification by recognizing archaea as a completely distinct domain equal to bacteria and eukarya. Second, it standardized taxonomy. It established rRNA sequencing as the gold standard for identifying and classifying microorganisms globally. Third, it advanced our understanding of extremophiles and origins of life. Studying archaea provides crucial insights into the harsh conditions of early Earth and how life might have begun. And finally, it laid the foundation for modern phylogenomics, providing the computational and genetic framework for today's evolutionary biology. These points show this wasn't just an academic change. It revolutionized how we identify microbes, explore life's limits, and even how we trace our own deepest ancestry. Here are the key takeaways from our session. The rRNA molecule serves as a reliable universal molecular clock, allowing us to trace evolutionary history across all life. The three domains, bacteria, archaea, and eukarya, accurately capture the deepest, most fundamental splits in the tree of life. The close evolutionary relationship between archaea and eukarya has fundamentally reshaped our models of cellular origins. Today, this classification system guides cutting-edge research in evolutionary biology, medicine, and biotechnology. These takeaways remind us of the core concepts, the tool used, the new map of life it revealed, and the ongoing impact this knowledge has across science. Thank you, Mootechie. Don't forget to like, share, and subscribe for more biology content. And that brings us to the end of our exploration of Carl Woese's groundbreaking work. By looking into the molecular heart of cells, he gave us a new map for the journey of life itself. We hope this deeper understanding of the three domains inspires your own curiosity about the living world. Thank you for learning with us.